Okay, question 2. In mammals, in the early stages of pregnancy, a developing embryo exchanges substances with its mother via cells in the lining of the uterus. So we've got exchange. At this stage, there is a high concentration of glycogen cells in the lining of the uterus. It's just context. There's nothing useful about that for question 2.1, which is describe the structure of glycogen. Very straightforward. I kind of wish more questions were not. This is very much AO1 factual recall. 2 marks. Again, 2 marks, it should be 2 bullet points. Look, I mean, like, you do not want to be writing this much space. Remember our CARBs, which is the acronym for analyzing a question. We've got command word and marks, describe, and 2 marks. The actual topic of this question is very straightforward. So this is the structure of glycogen. The key terms, obviously, we want to be talking about the type of, what type of glucose is it? Is it a polymer? Is it a monomer? What kind of bonds form it? What else could we be talking about for glycogen? Is it branched or is it unbranched? Is it soluble? Is it insoluble? That's more linking it to the function, but all these things can flick through your head to start with, and then you're going to pick out the ones that most relate to the structure, and you're going to then put them in a nice series of concise bullet points with a key term in each one. Yeah, short sentences. You can use bullet points or you can use full stops as bullet points, which is what I tend to do. Then at the end, we can self-check our answer. So carbs is command word and marks is C. A is the actual topic of the question, not what the face topic of the question might be. The review key terms is R. You've got bullet point answers, and then self-check is the S. The self-check, you can think, again, how can you add more key terms is generally what you're saying there. 2.2. During early pregnancy, the glycogen in the cells lining the uterus is an important energy source for the embryo. Suggest how glycogen acts as a source of energy. Do not include transport across membranes in your answer. Okay, so how is glycogen a source of energy? Some key terms here. You might want to maybe obviously say it's going to be broken down to form something. What is that something used for? What is the ultimate aim of that? Is to make ATP, because energy, you can read energy as being ATP. So that's going to be a key term in your answer here. I don't know exactly where that answer is going to go, but basically glycogen is a source of something, and that something can be used by a process in order to make ATP. Okay, so suggest and explain. So we've got to do 2 things, presumably for each marking point here. 2 ways in which the cell surface membrane of membranes, so it's the cell surface membranes of the cells lining the uterus may be adapted to allow rapid transport. Okay, so it's the cell membranes. My first thought here was maybe this could be villi or microvilli. Well, that's an adaptation of the cell, but not of the cell surface membrane. So what features of the cell membranes are involved in transport, and how might they be adapted for transport of nutrients? Probably I would avoid water and osmosis and aquaporins here, because debatable whether water would be a nutrient. So nutrient, we've obviously got glycogen as reference from the previous part of this question. How's that get into cells, for example? So let's, and for each marking point for these, you're going to have a suggest part and the explanation, the reason as to why that adaptation allows for the rapid transport of nutrients. So suggest and explain for each point. 2.4. In humans, after the gametes join at fertilization, every cell of the developing embryo undergoes mitotic divisions, not meiosis, mitotic division, before the embryo attaches to the uterus lining. The first cell division takes 24 hours. The subsequent divisions take 8 hours, so there's a difference there. After 3 days, the embryo has a total volume of 4.2 times 10 to the minus 3 millimeters cubed. What is the mean volume of each cell after 3 days? Express your answer in standard form. Show your working. Okay, so after 3 days, the embryo has a total volume of this. We need to know how many cells are present, and obviously then divide the total volume by the number of cells to get the volume per cell, or the mean volume of each cell. Okay, so how are we doing this? Well, 3 days? The first division, the first 24 hours, there's 1 division. The next 2 days, it's every 8 hours. So I'll just do a little bit of the calculator work with you here just to start pointing you in the right direction. So you've got 24 hours in a day, that's 34. So check your numbers that you're punching into the calculator as a first off. Let's actually pull this up here, it'll be easier. You've got 24 divided by 8, yeah, which tells you that during days 2 and 3, there are 3 divisions each. So you've got day 1 is first 24 hours, 1 division. And then the next 24 hours, you've got 3 divisions. And then the next 24 hours, you've got 3 divisions. So that's a total of 3 plus 3 plus 1 gives us 7 divisions. Okay, so each time you're starting with 1 cell. 1 cell, 1 division takes you to 2 cells. And then every division, what happens to those cells? So how many cells do you have is the first section, and then you've got to divide. Is this number going to get bigger or get smaller? If this number gets bigger, what do you do to what is this happening to the -3? If the number gets smaller, what happens to that little -3 there? So they are the skills that are involved in this. And obviously, if you get totally stuck, sometimes if you've not practiced this enough or you're not familiar with standard form, this could take forever. You could play around with numbers until the end of the earth. But if that's the case, mark it in the margin and come back to it. It's 2 marks, should take you a maximum of a couple of minutes, 2 to 4 minutes.